We will discussing diversity. Today we will discussing about diversity. In this video, I will discuss diversity, why diversity is important, and what kind of diversities you can find out. So video spokes on different organisms. They can prepare the diverse in their features. Well, so let's start the video. So introduction of microbiology. Introduction of microbial diversities. So microorganisms. Micro means the living things. So living any kind of organisms. For micro means are tiny, small naked eyes. It will be the see that. So organisms means for living things. So micro means tiny, organism living things. So another one, diversity. Diversity is a variety of types. So microbial diversities. This is a landmarks in microbiologies. So what kind of landmarks in the year? First of all, I told you. So introduction of microbiology is some of the authors. Louis Pasteur for Anton von Leeuwenhoek, 1687, first to see microbes with microscope. Leeuwenhoek. Pasteur 1864, proved germs cause disease plus milk pasteurization. So I tell you landmarks. So first Leeuwenhoek 1687, first to see microbes with a microscope, and second one Pasteur 1864, he proved germs cause disease plus milk pasteurizations. And third one Koch 1895, he proved specific microbe causes specific disease. And fourth, Watson and Crick 1953, bound DNA double helix structures also. So I tell you the landmarks. So next we will discussing about it. Microbial diversities: virus, bacteria, archaea, fungi, algae, protozoa. This kind of the microbial diversities. So first of all, how microbes are different. So there are three ways from your first one, safe. Three ways for microbial diversity. First, safe. Second one, cell wall, and food type. So safe for coccus, round, bacilli, rot, spirillum, spirillum. This is the safe. So this is also the bacteria. And another one, virus. That kind of virus are all living things. So there are the virus, the poisons. Virus is the poisons. So it is not a living cells, just DNA, RNA plus protein code, no nucleus, no cytoplasm, cannot make food. So virus. So living or non-living things. Okay. So next slides are discussing about bacteria. So bacteria so many shapes. So already told you diversities. Microbes are different universe of microbes. Especially all the bacteriums, there are potentially our body infectious agents. So Streptococcus pneumoniae is a round shaped, Clostridium tetani is a drumstick shape, spiral shape or spirocharts. There are the so many different type of the bacteria is arranged. And the microbe cell shapes also, when combined together in different shapes of structure. Different shape of structure under the microbes. So there are makes them put in one genome similarities that was created all the same times. Particularly diversities is you understand. Okay, and archaebacteria means a unicellular prokaryotes live in extreme environments. It is a methanogene producing methane. So nomenclature of microorganisms designed by Carol Linnaeus 1735, and Carol Linnaeus nomenclature is mentioned about archaebacteria and eubacteria. So many cell types, what are the cell type, and prokaryotic and differentiation and eukaryotic differentiations are mentioning about it. So the archaebacteria and bacteria, eubacteria, each one different cell shape. So this is the model of the origin of eukaryotes. For plasma membranes. So imaginations, you see that plasma membranes may have the formed the nuclear enveloped for. And next we are saying the three domain system, bacteria and archaebacteria and eubacteria. So what kind of the three domain systems. Okay. And next slides on a tree of living including archaebacteria and eubacteria. There are the different groups, archaebacteria and eubacteria. In this figure adapted for, so already told you microbial diversities, bacteria, fungi, virus, archaebacteria, and next I will go discuss the fungi. Okay, these the three kingdom classification is a old classification by earliest Haeckel classifications, the 1866. This is also there. So the each one classifications kingdom, Plantae, Animalia, Protista, and who is it in Plantae? Plants plus plant-like microbes. Plantae, plants-like microbes. And cell type, eukaryotic cell wall, make it own food. This is the cell type, Plantae cell type. For example, algae and cyanobacteria. Okay. And next animal kingdoms for Animalia, animals plus animal-like microbes. Cell type, eukaryote. No cell wall. Eat food. For examples, protozoa, amoeba and parameciums also. This is the examples. And next kingdom, Protista. Protista for this is the dustbin kingdom. Leftovers. Cell type, eukaryote, both plant plus animal features. What kind of examples? Yeast, fungi, all bacteria. And next kingdom also Protista. So this is the Plantae, Animalia, Protista. These kingdoms are three kingdoms. Three kingdoms to mix it up: bacteria, algae, fungi, protozoa, all in 1-2 kingdoms. Not accurate. Okay. So 5 kingdoms, 3 domain now. So from your timeline slides, 1977, discovery of archaea by scientists found in archaea look like bacteria, but DNA is totally different. 3 kingdom was not enough. Okay, thank you student. Thank you. So subscribed my channels. Next we will discussing about next slides. So any doubts, your